And welcome back to the Emerging Growth Conference. Next up, we have Pressure Biosciences, Inc. It trades on the OTCQB under the symbol PBIO and is a leader in the development and sale of innovative, broadly enabling pressure-based solutions for the worldwide life sciences and other industries. Please welcome its president and CEO, Richard Schumacher. Welcome, Richard.
Thank you, Anna. Thank you very much.
All right, the floor is yours whenever you're ready.
Wonderful. I want to thank everybody for taking time out of your busy days to join with us today and learn a little bit about Pressure Biosciences. First, before I begin, I'd like to read a little bit about forward-looking statements to remind everybody that things I say may be viewed as forward-looking statements. So please, before thinking about investing in a company, publicly traded company like PBIO, please do your homework. We are a leader in the development and sale of very innovative instruments, consumables, and testing services that all have one thing in common. They're all based on the use of very high or even super high pressure. Our customers are pharmaceutical companies, biotech companies, agriculture, food and beverage, cosmetic companies, and also academic institutions and government labs from all over the world. We have 29 issued patents that allow us to practice our inventions and also give us the protection that we need. As a young scientist many years ago in the Harvard Medical School system, I was working with a number of biological samples, viruses, bacteria, cells, human, animal, plant cells, normal, cancer, etc. And I was taught, like many scientists like me, thousands, hundreds of thousands of scientists all over the world, that the way to control the samples that we're working with in the lab is to use heat and cold to speed things up, slow things down, and use chemicals. All of these things worked and worked quite well. The problem is that when you're done with your experiments, you have to bring the samples back, the biological samples back to the initial stage of the initial temperature that it started at. And also you often have to take the chemicals out. Pressure is an amazing force of nature. Pressure moves at the speed of sound. Pressure can be very instantaneous. It's like turning a light on and off, turning pressure on and off. Pressure can do many of the things that we used to do in the lab, that I used to do in the lab with biological samples. It can do many of those same things, but it does them with much more exquisite control. And when you shut off the pressure, everything goes back to normal where you started. So it has an amazing number of significant advantages over using temperature and chemicals to modulate the biological samples that you're working with in a lab. Unfortunately, pressure really hasn't been used that much over the last few centuries in the lab, because up until now, no one's really developed a mechanism, an instrument system that could give you the pressure that you needed in a safe and very controllable manner. That's what Pressure Biosciences has done. We've captured high pressure. We've learned how to safely put high pressure into a system that can be used in the laboratory, that can be used with biological samples and other samples to help make things happen faster, slower, to stop things, to control the activities of different samples in the lab by using pressure, turning it on and off, rather than by using cold, heat, chemicals, and other physical methods. So first of all, let's begin to look at what we're talking about with pressure. Pressure is normally measured in pounds per square inch or PSI. If you look at the first slide that I have on, a lot of us are certainly aware that our tires run at about 30 to 36 PSI. Our espresso machines, maybe that we all have in our office at home, are running at about 100 PSI, which sounds pretty heavy duty, 100 PSI. If you look at the deepest part of the ocean, the Marianas Trench, Marianas Trench is about 7 miles down. And so you've got 7 miles of ocean. If we were holding this meeting at the bottom of the Marianas Trench, we'd have 7 miles of ocean on top of us. That would be about 15,750 PSI. Very, very heavy duty pressure. When you look at what Pressure Biosciences has done, what we've done in our instrument systems, we routinely go to about 45,000 PSI, and we have several instruments that go to 90 to 100,000 PSI. This is all hydrostatic pressure. We're pressurizing water and allowing that pressure to go through a test tube or another system that will then hold the biological samples that we're trying to manipulate. So we're controlling the pressure using hydrostatic pressure. And we are, in fact, going anywhere from 1,000 PSI to 45,000 PSI in our mainstay instrument, which you can see on the screen is the Barifold 2320 EXT. 45,000 pounds, almost 3 times the pressure you get at the bottom of the Marianas Trench. We get it instantaneously. We get it homogeneously. Everything in the pressure chamber is under the same pressure. And when we turn it off, it goes back to 14.5 PSI to normal pressure. Another instrument I'm showing here is the Ultra Shear technology. We'll talk about this instrument later also, called the Shear K45. This instrument also goes to 45,000. We've built a system that goes to 60,000 PSI in this system. This one is a system that combines high pressure with what we call high shear or cutting. So we combine, we make 1 + 1 = 10, we feel. So we're going to talk about Ultra Shear and the Shear K45 in just a little bit. So we've been working with high pressure for quite some time, and we've developed, in our 29 patents, we've developed 3 distinct pressure platforms: pressure cycling technology, or PCT, Barifold technology, or Barifold, and Ultra Shear technology, or UST. Pressure cycling technology is what it says. We cycle pressure. We turn pressure on and off. We have an instrument that will turn the pressure on and off in levels that you want it to go to. You may want to go to 20 or 30 or 42,500 PSI. You may want to hold it under pressure for 10 or 20 or 30 seconds. You may want to repeat it or cycle it 5 times in a minute or 10 times in a minute. You as a scientist make the decision. All of these things will affect the biological material that you're working with. Everything has its own pressure profile. Once you figure out that the virus you're working with, the bacterium, the cancer cell, the normal cell, the plant cell, the animal cell you're working with is able to be modulated in the way you want at a certain pressure, you dial that into the computer. You cycle the pressure, and you're able to get the effects of what we often in the past have had to use: temperature, heat, cold, physical means, and chemicals to get to that same effect. The second, Barifold, is a very different technology. This is a service. About 22% of all new drugs on the market, of all drugs on the market, are protein-based. They're made of proteins. 30 years ago, no drugs on the market were protein-based. Now about 22%, and that number is going to go up over the next 5 or 10 years. We make the proteins in the lab. When you make them in the lab, things can go wrong. 2 of the major things that can go wrong in the big vats where you're making the proteins is that the proteins can stick together or aggregate, or the proteins can be misfolded. Without getting into too much science, proteins are made up of components called peptides that overlap each other in a very, very specific manner. And when you're making it in the lab, it usually comes out perfect. But sometimes they don't overlap exactly right, or they aggregate. Pressure has been shown to disaggregate and to fix misfolded proteins. We own 8 patents in that area, like we own 15 patents in the pressure cycling technology area. Ultra Shear technology is our 3rd technology platform. The first instrument will be coming out at the end of the year. We have prototypes that we've tested and other people have tested in their labs. We have pictures and videos on our website to show what happens when you use the Ultra Shear technology. Basically, right now, the main purpose of Ultra Shear technology is to allow one to make a nano emulsion. I remember watching The Graduate back in the late 1960s, and the gentleman leaned over and told the graduate, you know, the key word is plastics. The future is plastics. Well, right now, the future is nano emulsions in a lot of industries. We have emulsions in our life everywhere. Milk is an emulsion, salad dressing. A lot of the oils we use are emulsions, cosmetics, pharmaceuticals, so many emulsions. And a lot of these emulsions need to be, are made into macro or micro emulsions. And if we make them smaller, if we make them into nano emulsions, it's been shown they can be more stable and become more soluble and therefore more bioavailable. So nano emulsions is the key word going forward. And using Ultra Shear technology, we believe we have the next generation instrument to make very stable, high quality nano emulsions. Here's a picture of some instruments that we have. You can see the 4 on the left are research instruments. And these are the ones that 3 of them go to 90 to 100,000 PSI. The instrument in the middle, the Barifold 2320 Extreme, is our workhorse instrument. We have 325 roughly instruments around the world in about 225 labs. And some of the key opinion leaders in proteins and other areas of science are using the Baricycler. We need to increase that number. It is our goal to increase that number into several thousand. But right now we have a foothold in many of the top labs throughout the entire world using the Baricycler. And it's being used for a number of different purposes, like discovering biomarkers and quality controlling drugs that are being made. The production instrument on the right, the Shear K45, we'll get to in a minute, but this is combining high pressure with high shear, which allows us to do some things that really we don't think have been done in as high a quality and as inexpensively as we can do with our instrument that'll be available by the end of the year. Our customers are who's who. As I mentioned, we have about 225 customers around the world. They're working in multiple different areas: biopharma, biotech, nutraceuticals, food, beverage, manufacturing. And it's a who's who a list of names of who our customers are. There's published over 200 papers and present every year at a number of meetings and conventions. So that where we were pretty much unknown as a technology 10 years ago, we're now very well known. And at this point in time, we need to build a sales force, which we really don't have, and start to proactively sell our systems. This is the first system. This is our PCT, pressure cycling technology, Baricycler Extreme. You can see the instrument on your screen on the left. There's a chamber on the right of the instrument, a computer on the left. You can dial in the amount of pressure, how long under pressure, and how many cycles of pressure. Every biological will have its own pressure profile. If it's a coronavirus, if it's a hepatitis virus, if it's a human cancer cell or normal cell, it'll have its own profile. You dial it in, and you and your colleague who's halfway across the world will get the same result of preparing the sample, because you're doing it the same way. This really can't be done in any other way of sample preparation in our estimation. You can see there's a number of consumables, but on the right side, we have a consumable that we developed, we invented, and we patented. This is a plastic tube that can withstand any pressure up to really up to 90,000 PSI, but it's used all the time in this instrument, up to 45,000 pounds per square inch. It is made out of a highly next age generation plastic, and the plastic will contort when it goes under pressure. You put a tissue sample, like a biopsy sample, in it, and you pressure cycle it back and forth. It'll break the tissue sample up in a very methodical way, so that when you're done, that tissue sample has now been broken up. The cells have been broken. The DNA, RNA, protein, and lipids that are inside the cell have been released, and the scientists can do their work. This is our mainstay instrument and the major area of our current revenue base. Barifold is a technology that controls the folding of the peptides in a protein when it's made. So as I mentioned, when a protein is made in the lab, sometimes it can be in disarray. Sometimes the peptides cannot be lined up exactly correct, or the proteins can aggregate. We own 8 patents, issued patents, and we have about 12 more pending that allow us to practice in the field of taking proteins that are either misfolded or that become misfolded and trying to correct them. Imagine $1 million of protein that a drug maker is making, and they find that they have a level of misfolded proteins that's unacceptable. We can try to save that $1 million or $5 million of protein. We own the patents. We have 6 customers by word of mouth that have come to us. 3 are multi-billion multinational companies. 3 of them are startups. One of the startups has now gone to, first time ever, our stage 2, which means that we're in the process working with them as they try to get their IND and get their approval to go and eventually go and market. We want to be their partner all the way up to market and while they're on market, and that our end game here is to have a piece of the pie when that product goes on market. Part of the reason is that Barifold helped to get there, so we'll be looking for a user fee or some type of a license fee while that product is on the market for hopefully an extended period of time. Shear technology, it's really a revolution in homogenization and bringing things down to a nano emulsion scale to a nanoscale. We're combining high pressure and high shear. And what we're doing is at a very controlled temperature, we are able to break, in the case of an oil and water nanoemulsion, the oil droplets from a glob of oil down to 50 to 60 nanometers, very, very smaller than a bacterium, very small levels of oil. Remember that the active ingredient in many pharmaceuticals and many pesticides, in many nutraceuticals and cosmetics is inside the oil. So if you can break that oil down to a very small level, it can be more controlled. It can be soluble in water, and therefore it can be, as if it's a drug, more bioavailable. So we're able to do this with our new instrument. We're working very hard to get the instrument out by the end of the year. We've had tremendous response from people we've talked to who continue to have problems. I remember buying vitamin water 5, 6, 8, 10 years ago, and realizing a day or two later that things are just shedding out of the vitamin water. I've seen this in so many different things, orange juice and many, many other juices and whatever. Imagine being able to make these into nanoemulsions where the oil is so small that it actually gets solubilized in water and stays in solution and doesn't come out for months, if not years at a time. This is the potential of Shear technology. Again, a little bit later, we'll talk about in more detail. There are 6 issued patents, including, and we expect many more, including a patent on the very special valve that we use, that is a nano gap valve. It forces the sample through a valve that turns the oil droplet into a nano-sized droplet. And we also have what's called a Barifold isolator, which allows you to clean it in place much easier than anybody else that's on the market now. Anybody in the market now, we don't know anyone that goes close to our pressures at all. And of course, we don't know anyone. We have a patent that would have the nanogap valve or the Barifold isolator. Here's a picture of an emulsion. Emulsion is when you have 2 or more liquids that you put together that aren't supposed to go together. Think about your vinaigrette, your salad dressing. Think about milk. Think about mayonnaise. These are all emulsions. Imagine if you could make that into a nano emulsion where they never separate. Imagine buying paint at the store, leaving it in the shed for a couple of weeks, going out to paint. Now it's separated out. Imagine it never separating out. So imagine your vinaigrette. You don't have to shake it like crazy before you put it on your salad. So when you mix oil and water, they don't go together. And what we do, and you can see the picture in the middle, shows the oil droplets that are just mixed in water. And you can see the pictures on the right of the creams and the food and the beverages, et cetera, and the pharmaceuticals that are made up of emulsions, where you're putting oil, usually containing an active ingredient, a hormone, an enzyme, an antioxidant in that oil. If body can't get out of the oil, body doesn't like oil. We try to get rid of oil as quick as we can. And that's why many products that are ingested, a lot of that product is excreted along with the oil. If you can make that oil droplet to a nano emulsion size, to a nano size, you have much better opportunity for the body's enzymes to break apart that oil and get the active ingredient out very quickly. In fact, it's known that nanoemulsions, the bioavailability of releasing the active ingredient is 90% or higher generally for a nanoemulsion, when it's 10% or less for a micro or macro emulsion. Here's a picture of 2 samples. It's the same product. The sample on the left is a macro emulsion. It's 10% oil that was put in water, and it was homogenized using a typical homogenizer. And if we let this sit for a while, it's going to separate out. But you can see it's very opaque. And on the right side, you can see that same material that we put through our Shear technology instrument, same material. We took those oil droplets that were 200 to 500 nanometers. We took them down to about 40 to 50 nanometers. They're so small, they become invisible. They essentially become soluble in water. So if those oil droplets contained CBD or contained a vitamin, it had a much better chance of being transported throughout the body, a much better chance of the body absorbing the active ingredient out of the oil. And that's one of the telltale signs of a nanoemulsion. Not always, but generally, as you see here, when you make the oil droplet so small, it actually is smaller than the wavelength of light, which means light goes through, it's transparent or translucent. And that's the same sample on the left that's gone through our system with 6 quick passes through our system, our prototype system on the right. This is a very interesting slide in that this is astaxanthin, which we've taken from Creel. Astaxanthin is an antioxidant. You can buy it in some stores. Astaxanthin is not very water soluble. So we took astaxanthin and we made it much more water soluble using it in our system. So we took it from a macro emulsion to about 50 to under about 50 or 40 microns, and we're able to actually make it so that it becomes much more water soluble. So we're very excited about this to be able to, because when you can use a smaller amount and get the same effect because it becomes more bioavailable, it becomes cheaper for the manufacturer to make, so they make more money, and it's better for us, the consumer, because we have to take less or we have to use less because it's more powerful, because it's more bioavailable, because it is a nano emulsion. So we're excited about our work with astaxanthin. Here's a cartoon about how our patented valve works. Basically, you can see the big green blobs of oil on the left that all contain the active ingredient. They're going through a very special valve. It looks very simplistic. It's far from simplistic, but we have been granted patents in the U.S., in China, in Australia, and Japan, and we expect European patents soon to give us protection for this valve. This is a schematic of how it works. Basically, all I want to show, it's like a 2-piston engine. There's 2 tanks that hold the solution, so it makes it very efficient, and it gets it to very high pressure in each tank. We release 1 tank at high pressure, and the fluid that's in there, it might be an oil containing CBD, an oil containing astaxanthin, it might be a vinaigrette, and we release it at basically supersonic speed, and we control the temperature, and it goes through this very exotic valve of ours. And when we do 1, 2, 3, 4 passes of this, we take something that's a macro emulsion, and not everything, but we've found that in most things we are able to turn it into a nano emulsion. Again, making it so that it's cheaper to make. You have to use less and get more effect from it, and so everybody wins. The manufacturer wins and the consumer wins. Here's some so-called water-soluble CBD that we bought on the market. We took this water-soluble CBD, and if it's water-soluble, we should be able to put it in water. Like if we took it in our body, it gets into water, and it should disappear, but it doesn't. We tried 6 different water-soluble solutions. All of them looked like toothpaste coming out when they got into water. Ours, we took that same material and we put it through our Shear machine, and you can see when we squirt it into water, it dissipates in the water, becomes essentially soluble in the water. There's pharmaceutical applications for Shear technology, which is why we're working day and night to get the instrument out. We have 3 different sizes of instruments. We have a benchtop instrument. We have a floor model instrument that most manufacturers be able to use. And then we're building a large manufacturing that if you're a very large manufacturer and you want to infuse a vitamin or a cannabinoid into a juice or into water or into a soft drink or beer, we have a very large system that we're working on with our partners at Ohio State University. Talk about that in a few minutes. So in the area of pharmaceuticals, about 50% of all drugs being made are very poorly soluble in water. So that's 50% that we can go to and help make them more soluble in water by making the oil that they're in much more soluble as a nano emulsion. New skin products, if we can make it smaller, it can go through skin faster and better. So the transdermal application of pain medicine and other medicines could be enhanced using nano emulsions. Beverages, imagine milk. Now there's all kinds of different milk out there, and there's a lot of companies that want to infuse milk and infuse other drinks. There's nutraceuticals that they want to put in. Again, this all can happen, and very effectively if the material is made into a nano emulsion. So it's a broad market appeal for the instrument that we're working at. Food and beverage, pharmaceuticals, eco-friendly agricultures, cosmetics, etc. So we've talked to pretty much individuals and groups in every category that I've just mentioned. And our partner, Ohio State, is actively talking to a number of well-known manufacturers, because in just a short period of time, in a month or maybe two, we'll be moving the large instrument to Ohio State to be used in their testing lab. Some of the top key opinion leaders in proteins and in other areas around the world are our customers. They've published a number of papers. They've written chapters. We're very excited as a small company to have the key opinion leaders in the world that are using our system and talking about our system. Please go on our website, pressurebiosciences.com. You can learn more about me. This is my fourth publicly traded company, so not my first rodeo. Very excited. It has as much potential, I think, as any of my previous three companies. And my previous three companies had a combined market cap of about $1 billion. So we're very excited about where this is going. We had a pretty tough year last year, as did most of us, both in business and personal. It really, really hit us hard. This year is quite different, as you're about to see. Recent accomplishments. What I'd like to say is our first quarter was one of the best quarters we've had in the last 3 years. We formed a food industry consortium with Ohio State. Ohio State is one of the top food and beverage academic institutions in the country. They have an entire laboratory for testing out new types of equipment for the food and beverage industry. We have a program with them, and we'll be bringing our instrument out to them. And we set up a consortium, and it's open to everyone. And we expect small companies and some of the biggest companies in the food and beverage in the world to join the consortium and have access to our Ultra Shear technology. We've announced collaborations with a company named Biosciences to help them with their drug. We're working with Leica, which is a Danaher company. Danaher is a $200 billion holding company. Leica is a 100-year-old microscope company. We're working with them in cancer diagnostics with our pressure cycling technology. We've been awarded new patents in the last few months. We have mentioned a customer that is now going to stage 2. And once they get to stage 3, they'll be ready. That's when they're in the FDA IND trials and clinical trials. And if we can be their partner to when they get out on the market, this is the payday for us. We make money working with them all along, but the big payday is when the drug gets to the market. And we've announced plans to release our instrument at the end of the year. We've also announced plans to acquire an eco-friendly agriculture supplier. I'll talk about that in just a few minutes. Value proposition is very straightforward. We control the actions of molecules. It's pervasive. It goes throughout so many different industries. We have 29 issued patents. The markets we're going into, that we're selling into, are all multi-billion-dollar markets. And we have some of the key opinion leaders in several of these markets already as our customers. So we're very, very excited about what this means to us. We have a pending agrochemical acquisition. We're hoping to close this in the next 2 to 3 weeks. We think that this will be additive to our revenue almost immediately. And we think that their revenue would be profitable revenue right from the get-go. They have a brand name, they have a base, and we're excited about this company and getting it closed as soon as possible. And the market opportunities there, many pesticides and many fertilizers are oil-based. We can use our Ultra Shear to make them more. They already have good products. We'll make them even better, and it'll save us money. And then the customer will have to use less to get the same effect. Finally, short-term drivers. Our PCT platform, we had a best year in 3 years. In the first quarter, the second quarter, the rest of the year is shaping up. We're having a terrific second quarter. I've already given that guidance. We expect the rest of the year to be a strong year. And we're going to be very proactive with Leica on the novel cancer biopsy analysis using our PCT instrument. Barifold, as I mentioned, we have 6 customers that by word of mouth have come to us. We have 1 customer that we're now working with their CMO, their contract manufacturing organization. So things are really moving. And again, 8 patents to protect us. Ultra Shear, we have talked to groups in many different industries. We have a partnership with Ohio State, who has a name going back many decades in the area of food and beverage. And depending agriculture acquisition. We think it'll close, and it's going to accelerate the use of UST. That is going to be a prime part of what we do over the second half of this year, is to make their already good products even better. So there's a lot of drivers that could help the company and help the stock in the short term. We came out of a pretty poor year in 2020, as we all did personally and on the business. It's quite different right now. We have a lot to look forward to. We're very excited about what the short term, let alone the long term future is for Pressure Bio. I want to thank you all for joining me, and I think I have like 1 minute, Anna, for questions.
Yes, you do have a lot of questions, Richard, but we'll make this really short. One quick question. You mentioned that the agrochemical acquisition could result in the addition of significant revenue, even profitable revenue from the start. So if the acquisition happens in July, how much additional revenue could be added to Pressure Biosciences by the third and fourth quarters?
Quite a lot. I know I've said that. I haven't really given it quantitatively. Let's just say the potential for that over the second half of the year would be to equal or exceed what we do now with all of the business we're doing with our pressure technology.
Great. Well, thank you so much. We're going to send you all these questions so you can answer them directly on your own time. You're just going to have to come back and extend your presentation because it's fantastic. Thank you for your time, Richard.
You're very welcome, Anna. Thank you.
All right. Thank you, everyone. Remember, we've got some more companies coming up. We're going to take a quick transition. Don't go anywhere. We're coming right back.